So we're just going to talk through the DNA extraction from strawberries today. You should find an LMS, a little sheet that looks like this that tells you how it happens. And on the back are some questions that you need to answer. This is what you'll do. You'll fill this in and then submit this on the LMS to me. So just the back questions you'll need to answer. But it's good to look to the front because this is a lab you can very much do on your own at home if you want. So I'll talk through some of the materials with you now and how we're going to do this. So we're just got a strawberry, a plastic bag, some gauze. You could use a coffee filter or cheesecloth or anything. That's just going to help us kind of strain out the different parts. A funnel and a test tube. You could just use a glass or anything like that you have at home. And then just a little mixture of kind of soapy, salty water. That's what we'll be using in the beginning. And the very end, we'll use rubbing alcohol. And the key for this is it's got to be very, very cold. If it's not cold, it won't work. So the first thing we're going to do is we're just going to take our strawberry. We're going to put it in our bag. And we're just going to try to break it apart into pieces. So I'm just going to start mushing it. I'm going to do it kind of gently because I don't want to break open my bag. And I'm just going to try to get rid of all the clumps that I have. So I'm going to keep doing that. And then we'll pause and come back when I'm done. Okay, so I've gone through and I've smushed my strawberry. There's no big pieces in it. It's very liquidy, and that's what I want. So I'm just going to go through now and add a little bit of my soapy, salty mixture. If you're making this at home, just pour in some water. Just do a little squirt of soap and a little dump of salt. It's really not that much. I'm going to pour a little bit in. And the point of this is the soap is going to break apart those cell membranes. The salt's going to help separate the proteins from the DNA because we're trying to get the DNA all loosened up on its own. I'm going to mix this together and then we'll come back when I'm ready to separate it. So I've gone ahead and I've gotten it all mixed together. I didn't want it to be too sudsy, so I just kind of gently rubbed it together to make sure that these things mixed. Now what I'm going to do is I'm going to pour it. I've taken my gauze and kind of spread it out and put it over the top of my funnel. I'm going to kind of hold it over my funnel, and the funnel is going right into my test tube. Because the goal here is to get as much of the liquid down to the test tube, but to leave all those kind of solid, clumpy parts of the strawberries at the top with the gauze. So I don't want any of those solid parts in there. So I'm just going to kind of let this drain out. You can see as it's going through, it's starting to fill the bottom of the test tube. Once I get the test tube about halfway full, then I know I've got enough liquid. Okay, so this took seriously like a minute to do. So we've got it about half full. There's my strawberry liquid. So now the important thing here is to look right along the edge. And I'm a little worried that this is going to be hard to see on my webcam, but we'll try the best we can. I might take some pictures if I don't feel like you can see it very well. But when I pour the cold, and this is just regular rubbing alcohol that you can get from anything, it's got to be ice cold. This came out of the freezer. So the goal here is that I'm going to pour this in, and then the rubbing alcohol will stay at the top. And then between the strawberry layer and the top layer, you should see the DNA come out, and it's going to look like little threads. Okay, and yeah, you can kind of see that. So I'm going to spin it. You can kind of see how there's like little threads there, and it's kind of got little oxygen. If I get a little bit of a flick, it will kind of mix it together. So you can kind of see, if I kind of tilt that right, that DNA floating around in there. It looks like little threads. Okay, now it's important to realize that a strawberry is not one cell. Thousands of millions of cells inside of one strawberry. And strawberries have lots of DNA in them, which is why doing this with, chicken wouldn't work out as well as like a strawberry and banana, because those cells happen to have lots of copies of DNA in them. Okay, so here's my DNA. Now you can use something at home if you've got if you're doing this in a cup or something like that, you could use like a knife. But what you can do is you can take, this is called an inoculating loop. We use it for bacteria mostly, but it's nice because I can actually pull the DNA out, because inside of here it looks really thin and delicate and fibrous. But when I try to pull it out, if I just kind of swirl this around in here, I can sometimes get the DNA to get hooked to it, and then I can pull it out. And it looks kind of weirdly snotty, actually. But that's kind of what it looks like because it's kind of all clumped together. So what I have here is not one strand of DNA, because one strand of DNA would be so tiny and thin, you wouldn't be able to see it. But I have hundreds of strands of DNA all come together. When you're looking at those end questions in terms of like, oh, when I'm looking at a rope, I can't see one thread from far away, but I can see lots of them together. Okay, so feel free to replay this as many times as you need, but use this information to go ahead and fill out your sheet. If you have any questions, as always, you can go ahead and email me or just link on there, ask questions on there right on my homepage.